Hello, good evening students. Today we are going to start with a rapid revision on the biological classification. I once again welcome you all for this interesting chapter on biological classification, and we are going to start on the rapid revision. So this chapter on biological classification is very important from the examination point of view, because we see in earlier topics of living world and all other kingdoms, this biological classification every student should know thoroughly, then only they can study exactly where each organism is placed, and we can know about how to know about each of these kingdom as well as their classification, everything in detail. So this topic forms the basis, and some amount of questions are being asked from this chapter. So this is going to be very important. So I want all the students to take up this and interact with me for the entire session, and we are going to have a quick, smart, sharp, rapid revision on this biological classification. Yes, so today we will just start with this revision. So coming to the biological classification, as we all know, the classification of the organisms, as you see, okay, they are actually, hello students, welcome to the rapid revision round. So coming on for the classification, like the systems whatever we do classify, they are actually classified on the groups into the kingdoms. Basically we say Planetae and Animalia. Then came the 3-system classification. Then it was Whitaker who gave the 5-system classification on this. So now we are going to see the most interesting about each kingdom, the classification, what are the organisms that they are grouped under that, and all that in this next 1 hour of our session. So the question 1, maximum nutritional diversity is found in the group Planetae, Fungi, Animalia, Monera. So where is the maximum nutritional diversity is found? So I'm just waiting. So please, if you are joined the session, please put the answers in the chat box. I'm just waiting for you all. Yes, you're all right. Yes, you're right. The answer is, yes, you've given the right answer. Yes, it is nothing else than the, yes, the answer is the Monera. Actually, you should know here one concept here, that the Whitaker, he was the pioneer in making the 5 kingdom, the classification. And among that, the mode of nutrition was one among them. Okay. So in this 5 kingdom classification, one of the characteristic which was grouped was the mode of nutrition. And in that, it formed one of the major criteria for the classification. Okay. So when you talk about this Monera, they have got the maximum. We all know that they are the prokaryotes, right? Okay. What about the Planetae? They are the autotrophs. Yes. What about the Animalia? They are heterotrophs, or they are saprophytes. What about the Fungi? The same. They are heterotrophic. But what about the Monera group? Here they are of both autotrophic or heterotrophic. And it can be again, they are again as chemo or photosynthetic, chemoautotrophic or photosynthetic. Whereas heterotrophic again saprophytic, or they are parasitic. So you can see the vast nutritional diversity is exerted in Monera rather than any of the other groups. So Monera is the right option. That is excellent, Tirisha. So Monera forms the right option for the answer, maximum nutritional diversity that is observed in the group. Organisms which display properties of both living and non-living. Yes. So what are the properties that we do see? Yes. What is the properties? Yes. The viruses, diatoms, lichens, and then here we have the bacteria. So which is the answer? So which is the answer? Yes, you're all right. The answer here is viruses. As we all know that these other three, they don't have these, they're all living, but which is exhibiting both living and non-living characteristics. We all know the viruses, okay, they are just a simple cell outside, but only when they get a host, they can replicate, right? Yes, excellent. Akshaya, Tirisha, excellent. Only when they enter, they can replicate. And without that, they don't. Yes, and that is how the viruses has got both the properties of the living as well as the non-living. Then we have the next question is the fungi. We are going to talk about the fungi. The fungi lack mitochondria, dictyosomes, plastids and endoplasmic reticulum. So what is the answer? So fungi, we all know the fungi, yes, they are the eukaryotes, right? Okay, so it has got a cell wall, but it will not have a cellulose. Okay, but what are the other things that you all should know? Yes, the answer here is it has got organelles. What are the organelles that fungi will have? Yes, you're all right. The answer exception is what? It does not have plastids. It does not have plastids. So what are the organelles here? It will have mitochondria. What are dictyosomes? They are nothing but the Golgi apparatus. It will have the endoplasmic reticulum, inclusions, vacuoles. Okay. So the Golgi apparatus, which is quite smaller. Okay. Yes, excellent students, excellent, you are all giving the right answer, excellent, you are all interacting. I am very happy to see this interaction, very good. So we will see why did we choose this plastids rather than all this. See, dictyosomes are nothing but they are the Golgi apparatus type of vesicles which is quite smaller, that is called the dictyosomes. And we have the inclusions, the vacuoles, everything are present, okay, except the plastids, the chloroplast. So the fungi lack plastids. That is the right answer. Okay, so you have to choose. See, when you are looking into the answer, think about the structure. See, what does all the fungi will have and what are the organelles that it doesn't possess. So only when you compare and contrast, then only you can come to a clear idea, because it's such a simple basic question. But when many of them, they do make a mistake by choosing any of the thing. But know the concept first, like what is about a fungi and what all it will have. So with that you can make the right answer, that is the plastids. Okay, it will have mitochondria, endoplasmic reticulum, Golgi, but what it doesn't have, this is the answer. Okay, excellent students. Next, coming to the other interesting thing that we are going to see is the infectious proteins. The infectious proteins are present in viroids. Okay, the satellite virus, C option is Gemini virus and D is pyrions. I'm just waiting for your answers. Yes, please think again. The infectious proteins, again I'm telling, the infectious protein are present either in the viroids or satellite virus, the Gemini virus or the pyrions. Now let us first see what are viroids. See, viroids are nothing but, okay, it will have only RNA and that protein coat will be absent. Okay, so those are the viroids. Okay, and it will infect only the plants. Okay, it will infect the plants, the viroids. What about the satellite virus? Yes, excellent students, you are coming with the right answer. I am very happy to see. Okay, the satellite. So what are the satellite virus? They are nothing but some subviral agents. They belong to the subviral agents. Again, they are also associated with plant disease. What about the Gemini virus? They are also like plant infecting viruses. They are plant infecting viruses, like you can see. But here it is a single strand DNA genome. Okay, so that you have to know. But what about the pyrions? The pyrions are nothing but they are the infectious protein. So your answer is what? The pyrion. So what is pyrion? The pyrion is an infective agent, yes, which is small proteinaceous infection particle. So these pyrions itself is like a pathogen, right? They are small proteinaceous infectious particle. Okay, so these pyrions are something that will cause this laughing death, which we call it as Creutzfeldt-Jakob disease, or there is another thing in a particular tribe people where we call it as Kuru disease. Okay, the Kuru disease. Okay, mostly it will affect the cerebellum, okay, and that is the cerebrum. So that is where. So here the proteins will be folded abnormally, and in this disease it results in some nervous degeneration. It's a sort of degeneration happening in the nervous system. So the pyrion is the answer. They are the infectious protein. Okay, so you are able to follow. Excellent. All have given the right answer. Excellent. Shall we move to the next? Okay. Here the next question is about the penicillin. So about the penicillin. See, I wanted you all to inform you saying that from penicillin at least 1 or 2 questions we are getting in a year. So I wanted you to concentrate on the topic of this penicillin. Okay. Any of the questions they do ask in a year. So as you see the question here, the total number of ascospores in ascus of penicillin is 8, 16, 4 and 2. Mostly biology students, when we see the numerals, we have the numer panic attack. Please do not have that feel. It is very easy where I will explain you all. Now you have to first understand that this ascus, which is the ascomycete, okay, the penicillin belonging to that. Okay, here it will undergo the sexual reproduction. Okay, so what does it have? In sexual reproduction, it involves the fusion of that archegonium with antheridium. I think you know this concept. So here it will undergo. Yes, excellent students. Jivita, Tirisha, Rohan, you are all giving the excellent, the right answer. Yes, let me explain and then we will come back to this. So ascomites do sexual reproduction and they are going to undergo, that is the meiotic division, and it is going to form what are called the unicellular ascospores. Okay, so what is that? Each ascus will have around 8 unicellular ascospores. So the answer is yes, it is nothing but 8. So penicillin is a member of ascomites, where an ascus forms the 8 endogenous ascospores. So that is the answer. Okay, so you please remember the 8 ascospores that is present in the ascus of penicillin. Okay. Yes, here we are coming to the next question about the pigment. So here also you can see in the examination point of view, they do ask about this blue-green algae, the cyanobacteria, where the question comes. Okay, so that is also important. So the pigment present in cyanobacteria is R-phycocyanin, R-phycoerythrin, C-phycocyanin, anthocyanin. See, actually the pigments, whatever are present, they are meant for capturing the light and harvesting the light energy and in the production of what you call in the process of photosynthesis. But here when you say about the pigments, okay, mostly we do classify the pigments as phycobilins. Okay, so the pigments of phycobilins, we have phycocyanin and then we have what is called the phycoerythrin. Okay, so under this you have to know that these phycobilins, they are water soluble pigments. Okay, so they are actually, we have the phycocyanin or phycoerythrin. The phycoerythrin is quite common in red algae, okay, and the phycocyanin, as the name gives here, you come across in cyanobacteria. But here see, the question is asked whether it is so we are now phycoerythrin will not be the answer or the anthocyanin. But what is the nearest? They have given whether it is C or R phycocyanin. So your answer should be closer to it. So let us see what is that. So the answer here is C. phycocyanin. So what is C. phycocyanin? Because the R. phycocyanin is present in the red algae. Okay. So which is more characteristic of cyanobacteria? It is the C. phycocyanin. So what is the pigment that is present in the cyanobacteria? It is the C. phycocyanin. Okay. So excellent students, Karthikeyan, Jeevita, Tirisha, Rohan, you all have given the right answer. Very good. Okay. So that should be the spirit. Okay. We'll move to the next. Which one of the following organisms is not an example of eukaryotic cell? So which is not an example of the eukaryotic cell? So we'll just see what is the example. Okay, the options are Amoeba protis, the Paramecium chordatum, Escherichia coli, Euglena viridis. So among this, which is not, again I am telling, whenever a statement or a question is given, please look whether they are asking for the right or the incorrect. So here is not an example of eukaryotic cell. So first the basic, what you mean by eukaryotes. This basic I hope children you all know. Okay, they are nothing but the cells which has got the, yes, the answer is everybody are giving. So the eukaryotes are nothing but which is the organism that, okay, which has got the membrane bound organelles and nucleus, membrane bound nucleus. Okay. So what we are going to see. So in our kingdom, except the Monera, all are what? They are eukaryotes. Monera is what? The prokaryote. Achha. So the amoeba, okay, the paramecium, they all belong to what? Under the protozoa, the protista. So they cannot be the option. What about the euglena? The euglena again, it also comes under the protozoid family, so that cannot be the option. So it is the E. coli. So we'll see the option. So E. coli is a yes, 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 excellent. So what is the answer? Very good, Rohan, starting from protista. So what is E. coli? The E. coli is nothing but it is belonging to the Monera. Yes, they are nothing but they are the gram-negative bacteria. They are the gram-negative bacteria, the E. coli. So that is the, so it is not a eukaryote. Excellent student. Okay. Yes, E. coli is the answer. Next, we will see the next question. The first life on earth, it consisted of provirus, bacteria, virus and protovirus. What is your answer? Yes. So there were many theories being put forth. So among that, there was a theory which was put forth by Tenkem, and he gave the theory what is called the, yes, the protoviral theory. He said that the first life which came on earth, so many have the immediate option going for bacteria or virus, but we call it as protovirus. So they say that the first is the genomic material of the protovirus form, and then what happened, that replication, the transcript, and that is how the basic genome of thing has formed, and that constituted the life, the base life, and from that only the other life would have originated. So this is your answer. Yes, excellent, Karthikeyan. It is the protovirus. Next question. Yes, excellent. Yes, excellent students. Now, which of the following organisms completely lack cell wall? Okay, completely lack cell wall. They are the smallest living cells known and can survive without oxygen. Microplasma, euglenoids, slime molds, all of these. See, one thing we have to know, that euglenoids or the slime molds, whatever, like they are not the smallest living cell. So you have to eliminate, or all of these. So it is the Microplasma. So the Microplasma is a free living prokaryote that doesn't have a cell wall, but they can survive without the oxygen. Mycoplasma, which we would have heard about it, right? The PPLO. So the Mycoplasma, they are the free living what? They are the prokaryotes. So which would be the answer? It is the Mycoplasma. Excellent. Excellent, Raj. Somebody has given the answer. Yes, Mycoplasma is the answer. Okay. So shall we move? So you have to understand, because many students, they will just see euglenoid or slime, they think it's smaller. So even smaller than that is the Mycoplasma. Very nano size, but they are free living. They don't need oxygen actually. That is the base concept. Next, coming to this, in lichens, the sexual reproduction is usually performed by. So we know what is lichen, right? The lichens, yes. So here it's actually the partnership between the algae and the fungi, right? So here, which is going to perform the sexual reproduction? Whether it is the algal partner only, the B option, fungal partner only, or both algal and fungal partners, or either of the two partners, like mutual, like either of them. So what would be your answer? Yes. Yes, what is the answer for the 10th question? Yes, excellent. I could see people answering. Yes, the answer is fungal partner. Excellent, students. Here the fungal partner only will perform the sexual reproduction, not the algae or both algae and fungal or either. So it is the fungal doing the sexual reproduction. So that is the answer. Excellent. Next question, we have the interesting one, what we see here. Yes, in which group of organisms, okay, the cell's walls form two thin overlapping shell which fit together. So in a group of organism, you have the cell wall that is going to form the two thin overlapping shell. Okay, it is going to just fit together. A option, slime mold. B option, it is the chrysophytes. C is the euglenoids, and D is the dinoflagellates. Which is the option? Yes, many people have started answering. Excellent, excellent, ma. Yes, the answer is, so before that you should know that the group of organism that do have that cell wall, which will have that overlapping shell, which will be like a soapy box, okay, because of the presence of that siliceous, mainly the silicon, so that fit in together. So it is the diatom which belongs to the characteristic chrysophytes. Yes, your answer is right, children, because in euglenoids, okay, instead of cell wall you find only a protein layer there. Okay, what about the dinoflagellates? Okay, the dinoflagellates is diatom is that belongs to chrysophytes. So the dinoflagellates, it is a stiff cellulose plates. So they will have a very stiff cellulose plates on the outer surface, whereas the slime molds, okay, they don't have cell wall at all. So which should be your option? It is your chrysophytes. So the chrysophytes, because of that siliceous material, it will have like a soapy box where they just overlap and they fit together, and that is how it is understood. So the diatoms, okay, the chief producers of the ocean, the chrysophytes, the family in which that is belong. So that is where we see. Okay, excellent. Yes, the pellicle. So the chrysophytes, the diatoms are unicellular. They have that two overlapping cell walls, okay, and they look like that microscopic glass boxes with shells. And this question they might ask you in the MCQ. So now once when the question has been put forth, like where you find these shells, the soapy box like, but the next will be, they are composed of that silica that is called the frustule, the portion, the shapes in which the shells they compose of, that is called the frustules. Okay. As next we are going to another interesting question. Okay, the bacteria, they are actually grouped under 4 categories according to their shapes. Okay, study the given figure and you are going to select the right option. So they have given A, B, C and D. So they have mentioned that in A you see the spores. See, whenever you are looking into a diagram, first you have to always view from left to right, look for the labeling is given or the labeling is just like that left. So always scan it in this manner. Okay, So look for A, B, then C and D, the zigzag manner. Okay, then only you won't make any mistake. Okay, so now look for the what they have given. Some spore is there, they are telling about some flagellum is there, it is looking in a characteristic shape. Now what would be your answer? Excellent students, everybody giving their right answer. Yes. So what is that? A, we see what is A, it is the bacilli. Okay, what is about the B? The spirilla, the spirillum. The C, the vibrio, the comma shaped bacteria, Vibrio cholerae. And then we see the cocci, Diplococcus. Okay, so it is the bacilli, spirilla, vibrio and cocci. So you have to match the right figure for the right terminology. So you can see the comma shaped bacteria, which is the vibrio form, the spiral shaped form, then the bacilli with the spore, and then the cocci shape. So this is how you are going to match exactly with whatever the figures given and the options there. See, look here, usually you can see the best way like about the vibriae, which is C, which is confirmed. So you can see the C, it would not be spirillate or the cocci. So the C, it can be either the B or C. So the next where you can check again back will be your D option. So again that also fits here, right? But when you come for this too, we see the spiral. So that is where it is going to give us the right idea and we have to choose the answer. Okay, excellent students. Now coming here for the another question, which is the kingdom Protista. Okay, the kingdom Protista includes life cycle showing sporic meiosis. B option, the life cycle showing zygotic meiosis. C option, the life cycle showing gametic meiosis, or it is whether both zygotic and gametic. We are talking about the kingdom Protista. Please bear it in your mind. So what does it have? So first you should know like what is gametic meiosis. So it's the formation of gamete where you have the haploid gametes formation. What about the zygotic meiosis? So it's quite common in the protist, yes, as well as in the fungi. So here they are going to have the haploid number throughout their life, and if at all they are going to have the mitosis, it is going to increase their haploid number of those structures which is present. What about the sporic meiosis? This is quite common seen in the algae and plants where the meiosis do take place in their spore formation. So what is your answer? Yes, exactly. The Protista group will show both gametic as well as the zygotic meiosis. So there is a gamete formation with the meiotic division and the zygotic meiosis, which I already said that they will have their haploid. Okay, so that is quite common in the protist as well as the fungi. So kingdom Protista will have both zygotic as well as the gametic. Okay, it will have both zygotic as well as the gametic meiosis. Okay. Next question here. Consider the following statement with respect to the characteristic features of the kingdom. So you are going to look into the various characteristics of each kingdom. Okay, so they have given 4 kingdoms like Animalia, Munera, Protista and Plantae, and we are going to choose the right option. In Animalia, the mode of nutrition is autotrophic. In Munera, the nuclear membrane is present. The C option is telling in Protista, the cell type is prokaryotic. And D in Plantae, the cell wall is present. So of the above statement, which is correct? Okay, so we are going to see as somebody giving the answer. Excellent, you are giving the answers. Excellent. So in Animalia, see, the mode of nutrition is autotrophic. No, it cannot be only one type. We have many type of modes of nutrition. Okay, so it cannot be autotrophic. The next is the Munera. So in Munera, which is already shown, it is a prokaryote, right? Okay, so whether the nuclear membrane will be present. Okay, and what about in Protista, whether the cell type is prokaryote? So what should be your answer? Excellent. The answer is in planet the cell wall is present. So the other options are all not near to it, right? They are all wrong. So which is the right option? Do you see? it is in Plantae, the cell wall is present. Okay, so that should be your answer. Okay, excellent. Everybody have given the right answer. Excellent, students. Next, coming to another nice interesting question, because I like this sort of questions where you need to trigger your IQ, think over and answer. See, when a freshwater protozoan, take an amoeba, is placed in a marine water. You take a freshwater protozoan, okay, and you are going to put it in a marine water, which is more concentrated, right, when it looks. What happens here? The contractile vacuole disappears. The contractile vacuole becomes bigger in size. The number of contractile vacuoles increases. The contractile vacuoles remain unchanged. See, always there is a concept of osmosis, I hope you all know. Suppose you are taking anything from the fresh water, you are putting in a highly concentrated, everything is a cell, right? So what will happen? Whatever the water is inside will move from the concentration, from the lower to the higher. So what will happen? It will try to shrink. So same thing happens here. Yes, all are giving the right answer. The contractile vacuole disappears. So that is the answer. It cannot be bigger. Might be if it is put on the other way around, it can become bigger in size. The number of vacuoles increase or unchanged cannot be the option, whereas the contractile vacuole disappears is the right option. Okay. Excellent, students. Jivita, Akshaya, Tirisha. Excellent. So now here comes another question. Viroids. So viroids, which we already saw related with one question there. So here viroids, they have a single-stranded RNA not enclosed by protein coat, single-stranded DNA enclosed by protein coat, double-stranded DNA enclosed by protein coat, okay, and none of these. So we are talking about the viroids. So what should be your answer on viroids? So we saw this question just before I was explaining you to about viroids. So what are viroids? They are nothing but, okay, the viroids are, okay, the viroids are, 1 minute. The viroids are actually, they are actually the thing which has got only the RNA. The protein coat is absent. If you remember this, it is easy now. I am giving you the clue. So what should be your option now? Your option here would be, yes, the answer is, it is a single-stranded RNA not enclosed by protein coat. So it is only the RNA which is single strand and the protein coat is absent. Yes, excellent, students. Many have given the answer. It is not the DNA nor the double strand and all. It doesn't have a protein coat. That concept you have to know. So if you have that, then you can choose this, and none of these cannot be. See, viroids, they are a group of pathogens which I told in the previous slide also. It is going to infect the plants that are smaller or simpler than any of the known virus. So they have that single-stranded either linear or circular RNA molecules without a protein coat. So that is very, very important. And this viroid does not produce proteins, as it does not have that initiation codon and all. So it can multiply by both RNA dependent and DNA dependent replication. So this is with, so mostly they infect the plant, okay, and that causes more of plant disease and all. So this you have to understand with that respect with viroids. Yes, excellent, students, excellent. Next, coming to the next question. The membrane bound organelles are absent in. Again here we are emphasizing you the very basic, the eukaryote and prokaryote. Just keep it in that classification and you can make out these answers. So the membrane bound organelles are absent in Chlamydomonas, Plasmodium, Saccharomyces, Streptococcus. So what should be your answer here? Yes, excellent. So it's a very, very basic question, the Streptococcus. Because you see all the other members like the Chlamydomonas, the Plasmodium or the Saccharomyces, they all are they are the eukaryotes. Okay. What about the Streptococcus? It's a prokaryote, so it doesn't have the membrane bound organelles. Okay, Streptococcus bacteria doesn't have a membrane bound organelle, so that is the option. Okay, so that you all should know. Next, which one of the following is not the basis of the 5 kingdom classification? What is the 5 kingdom classification? The Vitakas, right? We have to know about the Vitakar classification, the Vitakas classification, so which is very important. So Vitakar classification, which we say Robert H. Vitakar. So actually he is an American taxonomist. He was the one who proposed this 5 kingdom classification, which was based on the phylogenetic system. Acha. Now what is his view? He is saying which of the following is not the basis. So he had some 5 criteria for it, but he is saying that is not the base. Cell structure, body organization, reproduction, reserve food material. So what was his concept? We will see that and we will find out which is the odd one out. So the 5 kingdom classification is actually based on cell structure, so that is right. The body organization, so that could not be. Reproduction, yes. Then the mode of nutrition, so here, and the phylogenetic analysis. So they are not talking here about what? They are not talking here about the reserve food material, so that is the odd one out. So here you have to know that they talk about the complicity of the cell structure, they do talk about the complicity of the organism, they talk about the mode of nutrition, and we saw it was very maximum in the kingdom of Munira, which we saw in the first slide itself. Yes, and then about the phylogenetic analysis, about the evolutionary base. So that and even they say the ecological role of these organisms placed in the kingdom as producers, decomposers, etc. So that is how the Vitakar, he made a classification. So which is the odd one out? It is the reserve food material. Excellent students, everybody gave the right answer. Yes, excellent. Next, coming to another interesting question, the Aristotle. We all know about Aristotle. Aristotle classified the organisms, that is the animals, in 2 groups on the basis of presence or absence of RBC. The group which does not have RBC is Aneima, Eneima, Ovipara, Vivipara. So we all know Aristotle. He is a Greek philosopher. He has got so much vast variety in its contribution in various fields of our science. So he first classified the main basic kingdoms of planets and animalia. So once when he took the animalias into consideration, what did he look through? Whether there is presence of RBC or there is absence of RBC. Okay, so with this, what was the answer that has been given? Yes, excellent student. The group of animals which have RBC, they are called Eneima. The animals which do not have RBC are called Aneima. See, another thing which I can give you always in our medical thing, when anything put in front with A, we will say it is absent. Okay, like that. So if it is A, that means what? It's not present. So Aneima is lack of RBC. So Aneima which do not have RBC. What about Eneima? That which have RBCs. These Ovipara and Vivipara are not into the contest of relating with RBC. So your answer should be what? Aneima, which means that they are classified with the presence or absence. So aneima is what? The absence of RBC, and eneima is nothing but the presence of RBC. Okay, so that is how we do. Okay, shall we move to the next question? Excellent. Many students have given the right answer. Excellent. So the classification of these kingdom, first classification, it was made by Aristotle. Okay, so that is very important, that system. Then he made the system classification as a planet, the living organisms basically as plants and animals, and then he said that there is something with presence and absence of RBC, and he classified as aneima and eneima. Okay. Yes, we are going to go to the next nice question. Okay. So they are all very basic questions, but here you need to know the base concept. Whenever you are taking any MCQ, the first thing is listen or read the question, make your concept in the mind, then choose the right appropriate option. During favorable conditions, the bacteria mainly reproduce by budding, fragmentation, sporulation and fission. So what is the answer? So before going in for it, when you take a bacteria, they have mainly three methods. One, we say it is the fission, which we call the binary fission, and then we have what is called the sporulation, and then they can have what is called the sexual reproduction. Okay. So among that, now we are going to see what is the thing. So fragmentation cannot be there, nor the budding, which is not of characteristic of what? The bacteria. So we are narrowed down our answer to either sporulation or fission. Excellent students, everybody are giving the right answer. Yes, the answer is fission. See, you should understand what is the question asked. It is the favorable. So in favorable time, the bacteria would go multiplication by forming what we call the binary fission. That is, each cell are going to divide into two equal halves, and they are going to form what is called the identical daughter cells. They are going to have two identical daughter cells. Okay. Then the sporulation, the spores that is forming, that is not the option. So the answer is the fission, the binary fission. Okay. We'll go to the next option, next question. Mark the correct statement. Now we are going to see about the correct statement. Okay. So here we are going to see about, okay. So what is the correct statement? The phycomites. The phycomites include mushrooms, bracket fungi or puffballs. The second option, the mycelium of basidiocytes is branched and septate. Okay. The neurospora is used extensively in biochemical and genetic work. It belongs to the group basidiocytes. And then we have the morels and truffles, they are non-edible. So now you should know here about what is here we are talking, the bracket fungi. Okay. So it is not the phycomycetes. What are they? We are talking here about the basidiomycetes. Okay. So that cannot be the answer. The second, the mycelium of basidiocyte is branched and septate. Yes, that we are talking about the club fungi. Okay, we'll keep that in the option. The neurospora, it cannot be the member, it's not that member. And this morels and truffles, that non-edible cannot be. So which should be your nearest answer? Excellent students, the mycelium of basidiocytes, because they are branched and septate. Okay, they just grow in the soil, the branches and stumps of trees. So that is where the right correct statement, because the phycomycete is not the option. Here the neurospora is not of the member, nor the morels and truffles, they are not like edible, they are poisonous mushrooms. So which should be the thing? The nearest is the mycelium of the basidiocyte is branched and septate. Okay, we'll go to the next option, next question. Next coming to the another important, the mixed amoeba are formed in the life cycle of Physarum, amoeba, entamoeba, diatoms. So which should be your answer? So what is your nearest answer that you should give? Yes, what is that? The nearest, it cannot be diatom nor the entamoeba or what? We are narrowing it down. What is Physarum? They are nothing but the slime molds, what we are talking. Okay, so what we have are the myxomycetes. Okay, so they form individual cells which is called the mixed amoeba. Okay, so they are called the swarm cells which will develop with a flagella. So the answer is Physarum. So what is the answer? It is the Physarum, which are nothing but the slime molds. So the other options is not that, because you immediately jump into conclusion with amoeba. So that is not with the amoeba. So the life cycle of the slime mold is with the Physarum. Okay, so that you should all know. Okay. So next is the question, the paraflagellar body. The paraflagellar body of Euglena, it helps in locomotion, photoreception, reproduction, osmoregulation. So first when you see the Euglena, it's a eukaryote cell, right? And it's a single cell, and you have to know, first you should know about the paraflagellar, where it is actually with the base of the flagella. So it is mainly a light detecting structure. So what should be your option here? Yes, photoreception. So the paraflagellar is going to be with light detecting, means what? The photo, not the osmo, the reproduction or locomotion. The flagella is with for locomotion. But the answer for the Euglena, that excellent students, Karthikeyan, Rohan, you are all giving very good. So that is mainly meant for what? Mainly meant for the light detection, we say. So that is the answer. Okay, very good. Yes, excellent. Next question we will see, which is common to bacteria, mosses and fungus. So what is common to all these three? Their mode of nutrition, the presence of cell wall, they are autotrophic, their body organization. Now look into these three groups. Is that you think the nutrition will be same? No. Whether they will be autotrophic? No. Their organization of their body structure? No. So what will be the nearest option? The presence of the cell wall. It is the presence of the cell wall. Okay, so that would be your right option. So that is what is quite common and similar characteristic that you observe between the bacteria, mosses and fungus. Okay. Next we have another. Yes, excellent students, you have given the right answer. Yes, excellent. So it is the presence of the cell wall. Next there is another concept, what is called plasmogamy. Plasmogamy is a fusion of cytoplasm of two somatic cells of gametes, sperm with egg, two spores of different species of fungi, two vegetative somatic cells of different strain. So here we are talking about plasmogamy. So what is plasmogamy? It is actually a type of thing happening in sexual reproduction of fungi, okay, particularly the ascomycetes. That is the sac fungi, so it's quite common there. So what is there with plasmogamy? So here the parent cells, two parent cells, they will fuse without the nuclear fusion. So it is the cytoplasm of two somatic cells or gametes. So there will be the fusion of cytoplasm without the nuclei. So that is with the plasmogamy. Sperm with egg is what? It is the fertilization, syngamy. So this cannot be the option, the spores or anything. So what is that? It is the cytoplasm fusion, okay, but without a nuclear fusion. So that is called plasmogamy. Quite characteristic that you come across, okay. So that is very important, we come across. So they fuse, that is the haploid cells, they do fuse, very common in the ascomites, okay. Here comes the next interesting, match the following, okay. Here when you see here, they are asking us to match the genetic material with the virus. See, match the genetic virus that is given in column 1 with a virus that is in column 2. First you should understand what is being asked, okay. So here you see what are all they have given: double stranded DNA, single stranded DNA, double stranded RNA and single stranded RNA. Here they have given the name of all the virus: coliphage, circular virus, herpes, retrovirus, reovirus. Now we have to match them. So how are we going to sort out this issue? So now always when you see the double stranded DNA, if you are quite confident with those things, like we can very easily make it out. So usually many students, they will go option like this only. So I will just tell you, the option is B. See, the single stranded RNA, which we are very familiar, is with what? Is with the retrovirus. That mostly everybody knows. Next, coming to the double stranded RNA. the double-stranded RNA, the best example what we have studied is the reovirus. Okay. Now, single-stranded DNA. The single-stranded DNA is the circular, which we call the colipage. What about the double-stranded DNA? The most complex, the herpes simplex infection, the herpes virus. So, A is herpes, B is related to the colipage, C reovirus, and D the retrovirus. So, that is the option. So, B is the right answer. Okay. Next here we are going to see about the fungi here. Okay. So, coming to the 27th question. So, gill fungi, cup fungi, black mold, blue or green mold. So, here they have given a list of what is called a salmon disease, trama, penicillin, zygospore, apothecum. So, how are we going to match? See, in mycology, when the word trama comes, it is the inner fleshy portion of the mushroom. It is the inner fleshy portion of the mushroom. So, the basidiocarps, no, they will have that what you call the gill-like structure. So, they have the cap. Underneath the cap, they will have like gill-like structures like this. So, the gill fungi, okay, the gill fungi will always go in terms with the trama. Acha. So, the rhomba like that. What about the cup fungi? So, when I say the cup fungi, okay, it is with the apothecum. So, we have to know about the apothecum. So, the cup fungi is actually matching it with the apothecum. So, you have to know that the apothecum, like mostly very common in the ascomyces, what happens? They grow in a shape, particularly what is called the apothecum. Okay. Then that black mold, whatever is there, okay, which would cause the option here, the C4, which we say it is the zygospore. Okay. So, zygospore, the black mold, that is the zygospore. What about the green and the blue mold? It is the penicillin, okay, which we all will know. But what about the salmon disease? See, salmon disease is actually caused by water mold, which will cause a characteristic skin disease in salmons. So, that cannot form the answer in freshwater salmons. So, the answer should be gill fungi with trama, which will come characteristic of that basidiomycete, cup fungi the ascomycete with apothecum, okay, and then we have the black mold with zygospore, then this green and blue mold which we are quite known. So, that is with the penicillin, okay. So, that should be your answer. So, those apothecum actually we call that ascocarp, where there will be like widely opened saucer shaped bodies. So, they are that one which you should remember, okay. Then we will move to the next question. It is very simple. They have given two diagrams, two pictures, identify A, B. So, first when you look into it, look into the structure carefully, yes, and we are going to answer. We know that one is RNA virus, another one is a DNA virus. So, in respect to that we have to find out. So, what is A, what is B? So, very simple, this is nothing but the tobacco mosaic virus, yes, which has got that single stranded RNA. And what about here? We know that the virus which is infecting the E. coli, what is that called? The bacteriophage. So, your option should be the RNA virus, tobacco mosaic virus, and B, yes, excellent ma, the DNA virus, the T4 bacteriophage, yes, excellent. Next here coming to this, the association of fungi with roots of higher plants is called lichen, mycorrhiza, slime mold, neurospora. What is the answer? The nearest answer, it's very simple question, direct answer. So, we know that there is something called a symbiotic relation existing between the fungi, yes, as well as the higher plants. So, till you post the answer, let me give this explanation. So, the fungi help the higher plants in absorbing the minerals like phosphate, zinc, copper. The higher plants in turn provide the fungi with sugars. So, there is a mutualistic, a symbiotic relation happening. So, what do we call, like say for slime mold, Okay, we don't see such relations, nor the Neurospora. Okay, what about with lichens? It is between the algae and the fungi. So what should be your option? It is the mycorrhizae. Excellent students, excellent. Jivita, Akshaya, Rohan, Tirisha, everybody are giving the right answer. Excellent. Excellent. Okay, so you understood. So this is the relation. Next we are seeing another interesting question. What we call the cellulose is the major component of cell walls of Saccharomyces, Pythium, Xanthomonas, Pseudomonas. We will see which is the option here. Okay. See, before going in for this question, you see that what is the right option, whether it is Saccharomyces, Pythium, or whether it is the Xanthomonas or Pseudomonas. So what should be? The cellulose is a major component. So your option here should be the Pythium. Why did we choose Pythium? Because it is a phycomycete which has got the cellulose wall. What about the others? They will have the peptidoglycan cell wall. Okay, what about Saccharomyces? Okay, they have that chitin. So which is the one that is left here? It is nothing but the Pythium. So it is like actually they live on the plants, they are aseptate. Okay, and that will grow between the cell, they will put forth that hostria. Okay, that will invade the cells. Okay, so the answer should be Pythium. Okay, so cellulose is a major component of cell walls of what? The Pythium. So the Pythium is the phycomyte which has got the cellulose cell wall. What about the others like Xanthomonas, Pseudomonas? They are all eubacteria. Okay, and what about the Saccharomyces? It's chitin. So that you should know. Okay. Next, coming to the interesting question of it, match the fungi in column 1 to the respective class. So they were given the name of all the fungi, they are giving the class in which they do belong. Rhizopus, Penicillin, Histaligo, Alternaria. What about the class? Ascomycete, Basidiomycete, Deuteromycete, Phycomycete. Okay, so that we have to know. First we will see about the classes and then we will try matching out. What about these ascomycetes? They are actually the sac fungi. Okay, so what you see in the cases of those molds that would grow on the bread and all that, right? So that is with sac fungi, those Penicillin, Aspergillus and all that. Okay, what about the Basidiomycetes? They are the club fungi. So what is that with club fungi? So those mushrooms, okay, that would, you see that is with basidiocytes. What about the, actually the basidiocytes, they are the best decomposers of the wood and all. Coming to the deuteriocytes, like they are like something called a higher fungi, like they are actually the artificial class of fungi. Then we have the phycomycetes, the aquatic form of fungi which we say, the aquatic form. Now we will choose the answer. Okay, it's like we say it's something like unnatural form of fungi also. Okay, so what should be your option now? Yes, right. Your answer is yes, A. Excellent ma. Akshaya, Rohan, Sirisha, Jivita, you all gave the right answer. See, let me explain. See, we have the Rhizopus. The Rhizopus which I told you, yes, they are the phycocytes. So that is the something like aquatic or unnatural form. What about the Penicillin? Yes, the Penicillin is the ascomycete, which I said the Aspergillus, the Penicillin, the molds. Okay. What about the Eustalgia? It is a basidiocyte, it's a club fungi. What about the Alternaria, which is a higher fungi? Okay, so that is the deuteriocyte. So these are the options. So because when you look into it, those phycomycetes, no, mostly they are obligate parasite on plants. Suppose you take those phycomycetes, they will have what you call those aquatic habitat, and they form what is called the thallus and all, mostly saprophytic. So thinking about each class, their characteristic, like which group of that particular fungi will fall into these respective class, you should know. Then only you can match exactly the column whatever is given. Okay. Okay, so with this, I think I have completed the biological classification rapid revision. So hoping this it would have been very helpful. We had a very interesting discussion in biological classification in prospect with kingdoms, their mode of nutrition, the classification and various organisms that is being placed in this, and chapters with important weightage given we have discussed. So with this, I am just winding up with the biological classification session, and I hope to see you all in the next rapid revision sessions. Wishing you all the best. Okay, thank you students.